In this screencast, we are going to talk about enzymes and cofactors. Now, the catalytic behavior of proteins acting as enzymes is one of the most important functions performed by cellular proteins. Without catalysts, most cellular reactions would take place really too slowly to support life. With the exceptions of a few recent discovered RNA molecules that catalyze their own reactions, which we'll talk about in chapter 21, all enzymes are globular proteins. Now, enzymes are the most efficient of all known catalysts, and they can increase reaction rates up to 10 to 20 times that of uncatalyzed reactions. Now, two enzymes that you may run into just out there as over-the-counter products are lactate. This contains the lactase enzyme. If you're missing this enzyme or your body doesn't produce enough, it makes digesting dairy really hard. And so you can take this supplement, and it has that enzyme, and it will help you digest dairy. Beano also includes an enzyme that helps people digest vegetables a little better without the kind of pain of gas being produced. So these are two places that we see enzymes, and there's a lot of more digestive enzyme supplements out there. Those are just the two that I chose. So enzymes are just proteins that catalyze chemical reactions. And let's pretend like I can spell catalyze right. So catalyze chemical reactions. So I want to review catalysts just a little bit. So when we talk about a catalyst, what this is something that increases the rate of a chemical reaction but is not used up in the process. And so a lot of times what this looks like, for example, is we have two reactants, A plus B, and we add a catalyst. And A and B react to form substance D, and then we recover the catalyst. So the catalyst is unchanged throughout this chemical reaction. And so that means they can be used over and over again. Enzymes are true catalysts that speed up chemical reactions by lowering activation energies. So enzymes speed up chemical reactions by lowering the activation energy. Now, activation energy a lot of times is abbreviated as E for energy, or as a subscript A for activation. Now, what does kind of this activation energy mean? And so I just want to review this in case it's been a minute since you've talked about this. This is an energy diagram, and you have reactants here and products here. And what we find is that if this was an uncatalyzed reaction, it would have this huge hill of energy to go up before it turned into products. And so it would take a lot of energy to get over this hill. And that is called activation energy. So activation energy is kind of right here from the reactants to the top of the hill is E sub A. And then what happens with an enzyme is that lowers the activation energy. So that hill needed to react is not as high. And so that means the reaction can go faster. It's kind of like a bike. If you think about it, like the reactants here would have to bike up a really tall hill, and that would take a lot of time. But the catalyst makes the hill smaller, and so that activation energy helps the reactants turn into products. And then, of course, the catalyst is regenerated. Now, Enzymes are really well suited to their roles in three major ways. So there are kind of three characteristics of enzymes. They are they have enormous catalytic power. So okay. And so that just means that they are going to speed up those chemical reactions by lowering activation energies, and they allow those reactions to achieve equilibrium more rapidly. Okay, so basically, when we talk about enormous catalytic power, they speed up reactions by lowering the E sub A. That's what we mean there. Then the second thing they do is they are highly specific. In the reactions they catalyze. And a lot of times this highly specific is referred to as high specificity. And we're going to talk more about that on the next slide. Then the other thing that they do is they can, their activity as catalysts can be regulated. Now, so what does this mean? Well, even though each living cell contains thousands of different molecules that can react with each other in and really honestly almost unlimited number of ways, only a relatively small number of these possible reactions take place because of enzymes are present. So the cell really controls the rates of these reactions and the amount of any given product form by regulating the action of the enzymes. So we can summarize this by saying the reaction rate and the amount of formation of any product are controlled by regulating that enzyme action. So let's talk about specificity for just a moment. So enzyme specificity is that second characteristic that's important in life processes. Now, unlike other catalysts, enzymes are quite specific in the type of reaction they catalyze and even the particular substance that'll be involved in the reaction. So, for example, what do we mean by this? Well, if you go into a lab and you work with strong acids, for example, strong acids are going to catalyze the hydrolysis of any amide or the dehydration of any alcohol or whatever process you're looking at. However, if you look at this reaction here, this enzyme urease catalyzes only the hydrolysis of a single amide, urea. So this is urea, and urease will only catalyze the hydrolysis of urea. Now we're referring to this as hydrolysis, and I want to remind you, if you add water, that's a hydrolysis reaction. So just a brief review there. So that we've talked about is an example of absolute specificity, which is just what happens when an enzyme acts only on one substance. So that means one enzyme will catalyze and react with only one substance, and that's absolute specificity. So the enzyme acts only on one substance. Then, right, you can have relative specificity, where you end up having enzymes that catalyze the reaction of structurally related substances. So, for example, lipases hydrolyze lipids, and it could be all lipids, right? Proteases split up proteins, and that could be all proteins, or phosphatases hydrolyze phosphate esters, no matter what other functional groups are around, okay? And so that would be relative specificity, where the enzyme acts on structurally related substances. So, for example, proteases split proteins. Okay, I want to put an example here. Example, urease only hydrolyzes urea. That's it. That's all that does. Then you can also get some stereochemical specificity, and that is where an enzyme has the ability to distinguish between stereoisomers. So, for example, you can have D-amino acid oxidases that will not catalyze the reactions of L-amino acids. So, stereochemical specificity is when the enzyme has the ability to distinguish between stereoisomers, right? So, for example, right, D-amino acid oxidase will not catalyze the reactions of L-amino acids. Just won't happen. Okay. Now, one of the things you've probably noticed as we've gone through is I continue to talk about enzymes, and notice these enzymes all end in ASE. And so let's talk about how we name enzymes. And there's really two systems that we have. So, kind of historically, some of the earliest discovered enzymes were given names that ended in IN to indicate that they were proteins, right? So 3 digestive enzymes that catalyze protein hydrolysis are named pepsin, trypsin, and chymotrypsin. And these are all digestive enzymes. However, over time, people realized there was going to be a large number of enzymes, and so it made it helpful to adopt a systematic nomenclature. And that systematic nomenclature is known as the Enzyme Commission System, which is the EC system. And so this is just, right, our systematic way of naming. In the EC system, each enzyme has an unambiguous and often long systematic name that specifies the substrate. And the substrate is just the substance that it's acted on, and this will be one of the reactants. So when we talk about substrates and enzyme reactions, basically we have, if you think about traditional chemistry, we have reactants and then they go to products, right? Well, when we start talking about enzyme reactions, what we have here is we have some sort of reactants and an enzyme, and kind of another word for that other reactant is substrate. And then that goes to products. Okay, so the enzyme name specifies the substrate, which is just the substance that's acting on it. So on the previous slide, we had urease, and that was acting on urea. The functional group acted upon, and the type of reaction catalyzed. And those names end in ASE. So let's talk about some of these groups that these enzymes are grouped in. And so they're on the next slide, which is here. These are the 6 groups of enzymes. So you have oxidoreductases, and these are function in redox reactions, which are those oxidation reduction reactions. You have transferases, which transfer functional groups. So they'll turn one functional group into another functional group. You'll have hydrolases. These are hydrolysis reactions. Remember that these generally add water. That's an easy way to identify them. We have lyases. These add double bonds or the reverse of that reaction. They'll take away double bonds. We have isomerases, which will give isomerization reactions. So you'll go from one isomer to another isomer. And then you can have ligases, and these form bonds with ATP cleavage. Okay, so let's look at an example. So this is the hydrolysis of urea. So this is a urea molecule. And what we see here is that means the substrate is urea. Then we look and we see the functional group that urea has is an amide, right? And then the type of reaction is hydrolysis. Okay, and so the way that this goes is you do the substrate, and then the functional group, and then the type of reaction. So this would be urea. Okay, that's the substrate first. Then we have the functional group, which would be amido, and then hydrolase or hydrolase. Okay, and we'll do more of these in class, so we will practice this more. Now, more often though, we use common names. So these are usually shorter, they're a lot more convenient. Common names are just derived by adding ASE to the name of the substrate, or a combination of the substrate name and the type of reaction. So for example, on the previous slide, we talked about urea, amido, hydrolase as the EC name. Well, the common name there is just urea, which is the substrate, plus ASE. So it becomes urease, and that's it. So for example, another example, we have a lot of enzymes that are alcohol dehydrogenases. So from this, the ASE tells us it's an enzyme. The substrate is here. So that tells us alcohol functional groups are substrates. And then the reaction type is told here, that dehydrogen, that is a dehydrogenation, which is a removal of hydrogen. So throughout this text, we are really going to focus on using those common names, because that's what we see most often. Now, another thing we're going to talk about in the screencast are cofactors. So in chapter 19, we learned that proteins can be simple. They can only contain amino acid residues, or they can be conjugated with a prosthetic group present. Now, many enzymes are simple proteins, whereas many others function only in the presence of specific non-protein molecules or metal ions. So If these non-protein components are tightly bound to and form an integral part of the enzyme structure, they're truly prosthetic groups. But many of them are only weakly bound to the enzyme and are easily separated from the protein structure, and those are referred to as cofactors. So enzyme cofactors are non-protein molecules or ions required by an enzyme for catalytic activity. They are different from prosthetic groups because they are weakly bound to the enzyme. Okay. Now, when we talk about ions here, these metal ions that are cofactors are things like magnesium, zinc is a common one, and iron is another common one, right? So, for example, calcium's another one. There's a enzyme needed to curdle milk. It's renin. And it'll curdle milk only when calcium's present. Okay. Now, if these are not metals and they are organic compounds, then they're referred to as coenzymes. So coenzymes are organic cofactors, right, that are often derived from vitamins. Okay. Now, so this is organic. So if it's a cofactor, it can be a coenzyme or a metal ion. Metal ions are just, if it is a coenzyme, it is also a cofactor. Okay. Metal ions are just cofactors. There we go. So, but if it's an organic cofactor, it's called a coenzyme. Now, the protein portion of the enzyme that requires the cofactor is called an apoenzyme. So this is just the catalytically inactive protein formed by the removal of the cofactor from an active enzyme. So, for example, we have this inactive enzyme. It comes in contact with its cofactor, and then now we get an active enzyme. Now, coenzymes, like I mentioned, come from many vitamins. When we look at this, here is a list of vitamins, and then here are the coenzymes that they will go and form. These coenzymes bind reversibly to an enzyme and are really essential for its activity. So these bind reversibly, right? So, for example, one of the things that we see here is this particular reaction, and you can have lactate, and it can react with the coenzyme NAD+ when lactate dehydrogenase is present. And so when this occurs, then you get NADH and pyruvate. But lactate dehydrogenase is only going to react upon the lactate to when NAD plus is there. So it's really essential because it's actually the oxidizing agent. So NAD plus is one that we see pretty frequently. It's often aids in the transfer of the hydrogen from one compound to another. Right? And so what we see here is that the NAD plus comes along with the lactate dehydrogenase. It removes this hydrogen. There's a double bond here formed, and now that NADH has picked up that hydrogen. So that's an example of NADH working as far as their coenzymes are concerned. All right, so in Chapter 2, we're going to discuss several of these coenzymes that act as shuttle systems, and so we will circle back around to this later. In the next screencast, we are going to talk about enzyme activity, and as always, if you have any questions, please feel free to stop by my office.